Have you ever wondered how cells organize themselves to produce, process, and transport all the materials they need to survive? Inside every cell, there's a highly efficient system that handles these tasks like a well-oiled machine. This system is known as the endomembrane system. In today's video, we'll dive into the fascinating world of the endomembrane system, exploring the roles of the endoplasmic reticulum, the Golgi apparatus, and vesicles. By the end of this video, you'll understand how cells manage to coordinate their activities so flawlessly. Let's get started. The endomembrane system is one of the most important and dynamic parts of the cell. It consists of interconnected structures that work together to produce, modify, and transport molecules like proteins, lipids, and carbohydrates. Think of it as the manufacturing and delivery system of the cell. The main components of the endomembrane system include the endoplasmic reticulum, the Golgi apparatus, vesicles, lysosomes, and the plasma membrane. In this video, we'll focus on three key players: the endoplasmic reticulum, the Golgi apparatus, and vesicles. Together, they play critical roles in maintaining the cell's function and survival. To start, let's explore the first component of the endomembrane system, the endoplasmic reticulum. 1. Endoplasmic reticulum, the cell's factory. The endoplasmic reticulum, or ER, is a vast network of interconnected membranes that extends throughout the cytoplasm. It's like the cell's factory, where raw materials are processed into finished products. The ER is divided into two types based on its structure and function: the rough ER and the smooth ER. Rough endoplasmic reticulum, rough ER. The rough ER gets its name from the ribosomes studied on its surface, giving it a bumpy appearance under a microscope. These ribosomes are like miniature protein factories, and they are responsible for synthesizing proteins. Once proteins are made, they enter the rough ER, where they are folded into their correct three-dimensional shapes. Some proteins also undergo chemical modifications, such as the addition of sugar molecules to form glycoproteins. The rough ER plays a vital role in the production of membrane-bound and secretory proteins. For example, enzymes for digestion, hormones like insulin, and antibodies are all produced and processed in the rough ER. Once the proteins are ready, they are packaged into small vesicles and sent to the next destination in the endomembrane system, the Golgi apparatus. Smooth endoplasmic reticulum, smooth ER. Unlike the rough ER, the smooth ER lacks ribosomes on its surface, which gives it a smooth appearance. The smooth ER is involved in the synthesis of lipids, including phospholipids and cholesterol, which are essential components of cell membranes. It also plays a role in detoxifying harmful substances like drugs and alcohol, especially in liver cells. Additionally, the smooth ER stores calcium ions, which are crucial for processes like muscle contraction and cell signaling. Now that we've explored the functions of the endoplasmic reticulum, let's move on to the next component of the endomembrane system, the Golgi apparatus. Golgi apparatus, the cell's post office. The Golgi apparatus, often referred to as the Golgi complex, is the next stop in the endomembrane system. It's a stack of flattened membrane-bound sacs that resemble a stack of pancakes. The Golgi apparatus functions as the cell's post office, where molecules are sorted, modified, packaged, and shipped to their final destinations. When proteins and lipids are produced in the ER, they are sent to the Golgi apparatus in small transport vesicles. Once inside the Golgi, these molecules undergo further modifications. For example, sugars can be added or removed from proteins and lipids to form glycoproteins and glycolipids. These modifications are essential for the molecules to function properly. The Golgi apparatus also plays a key role in directing the molecules to their correct locations. Some proteins and lipids are sent to the plasma membrane, where they are incorporated into the cell membrane or secreted outside the cell. Others are sent to lysosomes, where they are used for intracellular digestion. The Golgi's ability to sort and package molecules with such precision is what allows the cell to carry out its complex functions. Without the Golgi apparatus, the cell would lose its ability to organize and distribute materials effectively. With the Golgi apparatus covered, let's now look at the vesicles, which are the cell's transport and storage vehicles. 3. Vesicles, the cell's delivery tracts. Vesicles are small, membrane-bound sacs that play a crucial role in the endomembrane system. They are like delivery trucks that transport molecules between different parts of the cell and even to the cell's exterior. Vesicles can be categorized into different types based on their function. Transport vesicles. These vesicles shuttle molecules between the ER, the Golgi apparatus, and other parts of the cell. For example, proteins synthesized in the rough ER are packaged into transport vesicles, which then deliver them to the Golgi apparatus for further processing. Secretory vesicles. Once molecules are processed and packaged by the Golgi apparatus, they are sent out of the cell via secretory vesicles. These vesicles fuse with the plasma membrane, releasing their contents into the extracellular space. This process, known as exocytosis, is how cells secrete hormones, enzymes, and other molecules. Lysosomes. Lysosomes are specialized vesicles that contain digestive enzymes. They break down waste materials, damaged organelles, and even foreign invaders like bacteria. Lysosomes are often referred to as the cell's recycling centers because they help the cell reuse its components. Endocytic vesicles. These vesicles are involved in the process of endocytosis, where the cell takes in material from its environment. For example, nutrients and signaling molecules can be brought into the cell using endocytic vesicles. The ability of vesicles to transport materials efficiently and accurately is what keeps the cell's activities running smoothly. They ensure that molecules reach their intended destinations without getting lost or damaged. Now that we've covered the main players of the endomembrane system, let's discuss how they all work together. 4. The endomembrane system in action. The endomembrane system operates like a coordinated assembly line, with each component performing specific tasks to ensure the cell's needs are met. Here's a step-by-step overview of how the system works. 1. Protein synthesis in the rough ER. Proteins are synthesized by ribosomes on the rough ER and folded into their functional shapes. 2. Lipid synthesis in the smooth ER. Lipids are synthesized in the smooth ER and transported to other parts of the cell. 3. Transport to the Golgi apparatus. Proteins and lipids are packaged into transport vesicles and sent to the Golgi apparatus. 4. Processing and sorting in the Golgi apparatus. In the Golgi, proteins and lipids are modified, sorted, and packaged into vesicles for delivery. 5. Transport to final destinations. Secretory vesicles transport molecules to the plasma membrane for secretion. Other vesicles deliver materials to lysosomes or other organelles. This seamless coordination ensures that the cell can produce, process, and transport the molecules it needs to survive and function. Let's move to its importance. 5. Importance of the endomembrane system in health and disease. The endomembrane system is essential for maintaining cellular health. Any disruption in its function can lead to serious diseases. For example, protein folding disorders. If proteins are not folded correctly in the rough ER, they can accumulate and cause diseases like cystic fibrosis. Lysosomal storage diseases. Defects in lysosomal enzymes can lead to conditions like Tay-Sachs disease, where waste materials build up in cells. Golgi dysfunction. Mutations affecting the Golgi apparatus can impair protein processing and lead to developmental disorders. Now here's a question for you. What is the specific region of the Golgi apparatus where proteins are sorted and packaged? Only a genius can answer in the comments below.